Chapter 4. Bacteria and archaea. Non-eukaryotic cells were the first to appear on Earth billions of years ago. All bacterial cells invariably have a cell membrane, cytoplasm, ribosomes, and one or few chromosomes. The majority have a cell wall and some form of surface coating. This is just a picture of a bacteria under a microscope, and this is another picture of all three of the different bacterials. You got bacteria, archaea, and eukarya. They're different types of cells. Structures. Specific structures are found in some, but not all bacteria. Are flagella, pili, fimbriae, an S-layer, cytoskeleton, inclusions, endospores, microcompartments, nanowires, and intracellular membranes. Bacterial cells look very simple and two-dimensional when viewed with an ordinary microscope. These are just some pictures of the structures. It shows the capsule, cell wall, cell membrane, ribosomes, chromosomes, nucleoid, flagellum, pili, plasmid, cytoplasm, plasma membrane, and there is some that are over here next to the flagellum on the red one, but my recording bubble is covering it. I can't see what it is. This is just a generic picture of a microscope that shows what a microscope looks like, and it shows that it has 3 or 4 different features to show enhancement of what you can look at under the microscope. It shows where you place your slide to look at bacteria, organisms, or whatever you have on your slide. Functions and activities of bacteria. In most bacteria species, each individual bacteria cell is fully capable of carrying out all necessary life activities, such as reproduction, metabolism, and nutrient processing. Some bacteria cannot act as a group. Bacteria sizes and shapes. Bacteria can come in many shapes, sizes, and arrangements. In terms of size, bacteria cells have an average size of 1 micron. Arrangements of bacteria. Coccus is when the cell is shaped like a sphere or ball. Baculus is rod-shaped. 2 types are blocky spindle-shaped, and the other is threadlike. When the rod is short or plump, it is called coccobaculus. Vibrio indicates there is a curve in the bacteria structure. Spirillum is spiral-shaped or curved. A rigid double helix twisted twice. Spirochete resembles a spring shape. The greatest variety and arrangement occurs in cocci. which can be single, in pairs, diplococci, tetrads, which is a group of 4, irregular clusters, staphylococcus, or in chains of a few hundred, streptococci. Cubicle is a packet of 8, 16, or more cells called sarcina. Rods are less varied in arrangement because they divide only on the transverse plane. Biofilms. Biofilms are microbial habitats with access to food, water, atmosphere, and other environmental factors. Biofilms also have serious medical implications. Biofilms. This is just a picture of the biofilm life cycle. It shows in order how a biofilm starts into the dispersion of the biofilm in a habitat, and then how the cycle just restarts. External structures. Appendages are common but not present on all bacteria species. Appendages that provide mobility are flagella and axial filaments. Appendages that provide attachment points are fimbriae, pili, and nanowires. Structures. Filament is a helical structure composed of proteins approximately 20 nanometers in length. Polar arrangement is when flagella are attached at one or both ends of the bacteria cell. Peritrichous is when flagella is dispersed randomly. Amphitrichous is flagella at both poles of the cell. Monotrichous is one single flagella. Lophotrichous is a bunch of flagella or tufts on the same side. And this just shows the different structures, outside structures of what would be on the bacteria. And it shows each one and how they would look. Chemotaxis is the tendency of organisms to move in response to a chemical gradient. Phototaxis is movement in response to light rather than chemicals. Conjugation is the connection of two different cells. Cell membranes and cell walls. Bacteria cells protect themselves with the S layer or glycolysis. The S layer is single layers with thousands of copies of the same singular protein. Slime layer protects cells from loss of water and nutrients. This is just pictures of cells and whether they have cell membranes or a cell wall. And this two specifically show prokaryotes and eukaryotes. Prokaryotes have a cell wall, cell membrane, ribosomes, capsule, flagellum, and nucleoid. The eukaryotes have cytoplasmic membrane, nucleolus, nucleus, nuclear membrane, cell membrane, endoplasmic reticulum, ribosomes, chloroplast, Golgi apparatus, mitochondrion, and it has something else, but I can't exactly see what it says. Gram staining. Gram staining identifies two generally different groups of bacteria. Gram-negative describes a cell with an outer membrane, cytoplasmic membrane, and a thin cell wall. Gram-positive describes cells with a thick cell wall and no outer membrane. And this is just a picture of gram staining and showing how it identifies two different groups of bacteria. This shows two different groups of bacteria under a microscope, indicating the purple and the pink. Inclusions. Inclusion bodies store nutrients away intracellularly. Some inclusion bodies carry condensed energy-rich organic substances, such as glycogen, with special single-layered membranes. This is just pictures of inclusion bodies and showing how bacteria store the food, water, nutrients that they need. And it's just showing what that looks like under a microscope. Archaea. Archaea differ significantly from other cell types because genetic sequences are found in rRNA, and they exhibit a unique method of DNA compaction. Archaea have unusual and chemically distinct cell walls composed entirely of polysaccharides. Archaea have incredible adaptations to environments that would be deadly to other organisms. Methogens convert CO2 and H2 into methane gas through unusual and complex pathways. The gas they produce collects in swamps and may become a source of fuel. Methane may also contribute to the greenhouse effect. Methogens. This is a picture that I just looked up and I googled methogens, and this is a picture that showed up. It doesn't really show anything different for me, so I'm going to take the word for it that is a methogen. Archaea. Archaea that adapted in very low temperatures are called psychophilic. Archaea that adapted to high temperatures are called hyperthermophilic. This is just pictures of Archaea that I looked up. This is Archaea under a microscope. This is also Archaea under a microscope. It just shows different pictures. Classification of bacteria in Archaea. The early method of classifying in Burgess was phenotypic, not genetic, traits of bacteria. This classification was based on shape, behavior, and biochemical reactions. These traits are still used extensively by clinical microbiologists. Classification is used to help organize microbes and study their relationships and origins.